Good afternoon to everybody.
Yeah.
Hi,
So, Carla, the Michael will be a little bit late for today's meeting.
Yeah.
Yeah. So we can start. He will join the meeting soon.
Okay. Okay.
Okay. So good afternoon. I make a short introduction for you. So good afternoon, everyone. It's our great pleasure to have Professor Carla Bosia for this today's seminar. And this is a serious seminar. In fact, it is the second one. So let's welcome Professor Bosia.
Thank you very much for the introduction. So I will share a presentation this time, just to avoid problems with the problems like the ones I had last time with my tablet. So I share. Okay. Can you see my? Can you see the presentation?
Yes.
Can you see my? Okay.
Yes.
Perfect. Okay. And just can you see my mouse moving? Little arrow moving.
Movie.
There's the little arrow that is moving along the slide. Can you see it?
Yeah.
The pointer. Can you see? Okay, just to know if I move it to. Okay, thank you very much. Okay, so today I will continue. Now I start. Today I will continue about talking about noise in genetic circuits. Some biological hints about it, because last time it was only theoretical. So what I will do is basically to recall the few concepts that we have seen during the previous seminar, which were essentially related to gene expression burstiness. So if you remember, I was talking about burst in protein production and burst also at the transcriptional level, but maybe I talk mainly about burst in protein production. And then I will put them on some experimental ground. So I will present you two seminal papers in which actually protein bursts were observed for the first time at the single cell and single molecule level. I'm not sure. So, and then after that, I will introduce the idea of transcriptional bursting. That will be just an introduction, because I'm not sure I will have the time to go deeper into the experimental part, but for sure I will talk about it next time. Okay. So and then first of all, I will recall the concepts that we have seen last time, because last time in the end I realized that probably I ran too fast. And so I also put here some of the equations that we have seen, so that they are present in the presentation with all the references to which I refer to, so that then I can share with you the presentation so that you have it. The first part, just to have an idea, there is this nice tutorial by Matthew Scott that you can find online. It's a tutorial he did for a summer school, which is called Tutorial in Genetic Circuits and Noise from the 2016. And there you can find basically all the concepts that I told you last time. So last time I began with telling you that we could somehow translate the idea of gene expression into mathematical with a mathematical formulation, in which we obviously we simplified, say, all the processes that happens in a real cell through effective parameters. And so here, for example, you see that there is a gene which is transcribed into a messenger RNA proteins with a given rate alpha m. Then the messenger degrades with a given rate, or can be translated into protein, and then the protein can be degraded. And then we can somehow formalize this idea, appealing to the mass balance, and basically writing down some ODE, some ordinary differential equation that describe the system. So here we have seen that we could write the variation in time of the messenger RNA of the protein as a function of the parameters present in the sketch shown here. And then what I told you is that most of the time we are interested in studying the steady state of the system. And so we just equal to 0 this equation, and we evaluate the amount of messenger RNA molecules and the amount of proteins at the steady state. Okay, and then this is for a unregulated gene, so for a constitutive gene expression, so a gene that is simply transcribed and translated. Then, of course, on top of this description, we can have regulation. So any gene regulation, and here you see two cartoons in which there are schema of the promoter activity, which can be either repressive, like a repressive function is G R of R. R stands for repressive, or an activating function like this one, GAR for activating action. So in both cases, the transcription factor binding dissociation constant is called KR for the repressive function, or KA for the activating function, and omega is the fold change due to the regulation. So basically, it's the capacity of this function. Having this in mind, then we can describe more complex circuits, like, for example, an autorepressor, which is the one that we have briefly seen last time, in which we have a protein, the repressor R, that represses its own transcription. Here is a cartoon in which, and this is shown in a more in a less, let's say, a bit less schematic way, okay, in which you see that M is the messenger RNA. Again, it is transcribed with a given rate, alpha M, exactly as before. But now there's also a function, which is a function of the repressor R, which is exactly this kind of function here. So basically, the transcription of the messenger RNA is repressed by a function that depends on the amount of the protein repressor itself. And then here there is the degradation term, and then for the protein repressor R, we have its translation rate times the messenger RNA molecules amount minus the degradation rate times the protein. Okay, so in this way we can, just appealing to the idea of this function that describe the promoter activity, we can describe many circuits. So the idea behind these functions that describe the promoter activity is essentially that the binding of the transcription factor to the promoter, so the DNA protein interaction happens quite fast. So basically the promoter is almost always active. That's the idea behind the possibility to describe the promoter activity function with a hill-like function like these two. And this is something that we have to keep in mind, because also in what follow, in what follow, when I will talk about Barsiness in protein expression, I don't refer to to the activity of the promoter. But instead, when talking about bar senescence transcription, so in the production of RNA molecules or RNA molecules in general, this idea of fast binding and unbinding to the fast activation of the promoter does not hold anymore. So in that case, we should really think about what happens when a transcription factor binds to the promoter, if and there the timescales are relevant somehow with respect to the other timescales within the system. Okay, then last time and I told you that when we are in presence of small reactant pools, what happens is that individual reactions produce great changes in the total population. So at that time, the deterministic description is not, say, useful anymore. And so these reactions are more properly described probabilistically, so with a probabilistic approach. So the deterministic rate constant K plus or K minus, say, the rates that we wrote down before correspond to the average rate of the reaction. While in the reality, reaction events require reactant molecules that needs to come together with enough energy to form the product. Okay. So at this point, the true reaction rate will be roughly distributed exponentially about their average. And so at this time, what we need to use is a chemical master equation to estimate the effect of these fluctuating reaction rates on the overall evolution of the system. Okay, so the key concept at that point was that when we are in presence of small reactant pool, we need to call in the game somehow the master equation in order to estimate the effect of fluctuating reaction rates. Okay, and then okay, there are many reactant molecules. For many reactant molecules, the species concentration evolve both continuously and differentially. Okay, and here there is a sketch where C is the concentration of a given reactant molecule in time. But then, okay, just to repeat the concept, when small numbers of reactants are involved, the concentration evolves stepwise. So here you see the same concentration as a function of time when C is equal to 10, 100, or 1000. As the concentration increases, then the behavior of this compound, the concentration of this compound C is similar to the behavior of many reactant molecules. But when the amount is very small, is very little, then there is a stepwise progression of the trajectory, as you can see. So what happens is that repeating an experiment many times, we obtain some repeatable average behavior that conforms very nearly to the deterministic description, as you see here. And some envelope here around the average that accounts for the fluctuation in all the individual trajectories. Okay, so this is just to somehow visualize this idea of fluctuations that was thrown in the that was thrown on top of this of the deterministic equation that I show you in the beginning. Okay, so what we can imagine is that we have this somehow mean behavior, which is usually well described by the deterministic equations, and then we have say a distribution of values around it that give us an information about the fluctuations in time. So how much we could deviate from the mean behavior. Okay, and then at this time, okay, to describe the system, say to take into account the fluctuations, we need to recall instead of the mass balance, the probabilistic balance. And so it was right at that time that I introduced you the master equation, where I recall the idea of P of NT, with P is the probability of being in a given state N, where N is a vector, which is the inventory of the system. So basically N tells us that we have N1 molecules of species 1, and 2 molecules of species 2, and 3 molecules of species 3, and so on and so forth, at a given time t. And then probabilistic balance, exactly as for the idea of mass balance, is given by flux in minus flux out somehow. And so we have the probability of moving from a neighboring state minus the probability to moving to a neighboring state in a given interval of time, in a given period of time, dt. And then basically raising this in terms of transition probabilities, and then summing over all the states and taking the limits in time for delta t that goes to 0, what we get is exactly the master equation here. Okay, it's exactly this master equation, which is then quite easy to write when we speak about genetic circuits. So what I show at that point was exactly this. So yeah, just to write down the equation, imagine that now we are interested in a system in which we only have transcription and degradation of a messenger RNA molecule. Okay, so our state N is the state N here that I was recalling before, is just M, represented here by the amount of molecules of messenger RNA molecules M. So we can go from the state M minus 1 to M and vice versa, or from N to M plus 1 and vice versa. And so then we have to write, say, the probabilistic balance, so this delta P being particular state, which is basically terms given by the gain. So how we arrive in the state M. So basically we arrive in the state M with a rate alpha M, okay, from the state M minus 1. So we have alpha M times the probability of being in the state M minus 1 at time t, okay. And then we can arrive in the state M also from the state M plus 1 with a given rate beta M, which is basically the degradation rate, this one. Okay, so beta M times the amount of molecules that we have in the state M plus 1, which is M plus 1, times the probability of being in that state, M plus 1 at time t. Okay, and then we have to subtract the loss term. Okay, so the probability of escaping from the state M, which are basically given by beta M times M times the probability of being in state M at time t, plus the probability of escaping from M in order to arrive to M plus 1, which is again given by the transcription rate alpha M times the probability of being in M at time t. Okay, then you can simply adjust the terms. Again, we're most of the time interested in the steady state of the system, and especially on the first two moments of the distribution at the steady state. The first two moments means the mean and the mean square, which can then be rephrased in terms of the variance of the system. And to do this, what I recall you was the moment generating function approach. Okay, so we recall to the moment generating function, which was this function f of zt, where z are putative variables, one variable z for each variable that we have in the system. So in our case, it's just one variable, which is m, the amount of messenger RNA molecules. And this generating function is defined as follows. So it's the sum over all the m that goes from 0 to infinity of this putative variable z to the m times the probability of having m molecules of the species m at time t. Okay. And then the approach is the follow. So basically, we need to rewrite the master equation, which was written in terms of the probability P, in terms of the moment generating function. So we simply rewrite the master equation in terms of the moment generating function F. Then there are a few properties that we need to recall about the moment generating function. So the fact that when it is evaluated for z equal 1 is z equal 1, this f is equal to 1, and this is exactly the normalization condition that we had on P. And then the other property that we need to keep in mind is that, which basically gives the name of moment generating function, is that by deriving this f with respect to the putative variable z and evaluating it in z equal to 1, we obtain all the moments of the probability distribution P of M of t. Okay, so here, for example, we derive one time with respect to z, we evaluate in z equal to 1, and we get the mean value of n, so the mean, the first moment. We derive 2 times, we evaluate with respect to z, we evaluate this derivative in z equal to 1, okay, and then we get the second moment, and from this we can move forward and so recover an expression for the variance, an expression for the means, and then from there having an expression for example, the noise in the system, the fluctuation, and so on and so forth. Okay, then in a case in which we have only one messenger RNA molecules that is transcribed and translated, what we found is that basically the variance sigma square is exactly equal to the mean. So it's simply given by the ratio between the transcription rate of the messenger RNA and its degradation rate. So the ratio between the variance and the mean is called fun factor, and is one possible measurement of noise in the system, which in this case is equal to 1, as you can see. Fun factor equal to 1 is typical of Poissonian process, of birth-death processes. And so if you measure the Fano factor, depending on how it is far from 1, the Fano factor, you have an idea of how far your system is from a Poissonian process. Your process is far from a Poissonian process. Okay. Another measurement of noise, which is adimensional, is the so-called coefficient of variation, CV, okay, of M in this case, so the CV of the messenger RNA molecule, which is simply given by the ratio between the standard deviation of the system and the mean amount of molecules, again at the steady state, okay, which in this specific case of a Poissonian process, of our Poissonian process, is given by the ratio between, it goes as 1 over the square root of the amount of molecules. So it scales as the square root of the amounts of molecules that you have in your system, okay? And then by repeating exactly the same reasoning for a system with an unregulated messenger RNA and its translated protein, so now we have 2 species, M and P, the protein, we can also get information about the first 2 moments of the distribution with respect to the protein amount. So we have the mean value of the protein and actually the variance, and then the standard deviation, and so a measurement of the CV, the coefficient of variation for the protein. Okay, and what we get at this point is very interesting, and it's right now that I introduce the concept of barstiness in protein expression. Okay, so the mean value of P of the protein is given by the product of the amount of messenger RNA molecules that you have at the steady state times the production rate of the protein, so the translation rate, alpha P divided by beta P, so the degradation rate. Okay, by expanding the mean value of the messenger RNA at the steady state in terms of what we found before, we found that P is equal to the product between 2 ratios. So transcription rate divided by the degradation rate of messenger times the ratio between translation rate divided by degradation rate. And if shuffling this, we can see actually that this is given by the product of two quantities. So on one side, we have the so-called burst frequency, which is called A, usually in the papers that refer to burst frequency and burst size, which is basically the ratio between the transcription rate of the messenger RNA divided by the degradation rate of the protein. So basically, this is the mean amount of bursts of protein production that you have in a given period of time, which is now written in terms of the protein half-life, so the protein degradation rate. And then we have another term, which is the bar size of the protein production, which is given by the translation rate of the protein divided by the degradation rate of the messenger RNA. So basically, the bar size tells us that each messenger RNA molecules that is produced is amplified by a burst which has exactly this size, alpha p divided by beta m. Okay, and what follows, I'm going to concentrate on these two terms, burst frequency and bar size, that were actually and surprisingly measured experimentally. So proteins are produced in bars, which has a given frequency, specific frequencies depending on the processes, and specific amplitude. And the other very interesting thing is that if we evaluate the CV of the protein, we make all the calculation, and then let's say that we assume that proteins are more stable than messenger RNA, which is usually the case, so that beta M, the degradation rate of the messenger RNA, is much bigger than beta P, the degradation rate of the protein. What we find is that the CV of the protein, it goes as 1 over the mean amount of proteins, and okay, the steady state, times 1 plus B, the bar size. Okay, so really the noise somehow of the protein depends on the size of the protein bars. Okay, and just to reason a bit about it, imagine now which is the power of different regulators that can interact with messenger RNA or proteins in terms of somehow controlling the noise of protein production or messenger RNA production that we will see later, just because they could somehow simply controlling something that has an effect on the translation rate or the rate of the messenger has a great impact on the level of the noise of the protein, because it means to control the bar size, the protein bar size. Okay, and there are different regulators that can control this. One of them probably that belong to the same family are the micro RNAs, for example. I will talk about them in the next seminars. Okay, actually they can control the translation rate of proteins or degradation rate of the messenger RNA. So there's this idea that that really they have an impact on the noise control of protein production. Okay. So a major source of stochasticity in gene expression is due to the burst in protein production. In fact, proteins have been observed to be produced in brief periods of high expression intensity, followed by waiting periods. And this was measured experimentally in this paper by Kai and Yu, which are the paper that I'm going to present. And the idea is that this sparseness is mainly due to the fact that during the lifetime of a single messenger RNA, there are several proteins that can be produced. Just imagine that we can have many ribosomes attached to the same messenger RNA proteins, and so many to the same messenger RNA molecules, and so many proteins that can be produced at the same, more or less at the same time, from a single messenger RNA. And this is exactly the burst. Okay, so during the span of a lifetime of a messenger RNA, we can have many proteins that are produced. Okay, and therefore fluctuations at the transcriptional level can be amplified by the translation of a large protein burst stemming from a single messenger RNA protein. Okay, and so we recall like as the product between burst frequency, transcription rate with respect to the time scale set by protein degradation. Then usually, and we will see this in the experimental paper, if the proteins are very stable, then this burst frequency is given in terms of cell cycle time, and not times, is given in terms of cell cycle unit of time. Okay, so the dilution of the proteins due to the cell cycle. And here we have the bar size, which measures the amplification of the transcription noise by translation. So each run transcript is amplified by exactly this ratio here, alpha p divided by beta n. So the surprising thing was that we could observe burstiness experimentally. Researchers, and then I'm going to present you these two papers in the late 2006, so almost 15 years ago. They show in these two seminal papers appear in Science and Nature, more or less at the same time, that it was possible actually to measure protein burst. So now I'm going to talk about these two papers. So the first one, the title is Probing Gene Expression in Live Cells, one protein molecule at a time. So basically, what the author did in this work was to measure gene expression at the single molecule level by real-time monitoring of protein production in live cells. Okay. And here you see the experimental design that they design. So in which essentially there is a gene fused with a fluorescent protein, Venus. The gene is this TSR gene. And this gene has been cloned substituting the LAC-Z gene within the LAC promoter. So this gene now is expressed under the control of the LAC repressor, which binds to the LAC operator on the DNA. Okay, and then, so basically they substitute the LAC-Z gene with this TSR fused with the Venus fluorescent protein. Okay, and now what happened is that this gene TSR plus Venus is transcribed only when the LAC repressor is not attached to the LAC promoter. Okay. What is this Venus molecule? Venus is a YFP variant. Okay, so it's a yellow fluorescent protein with short maturation time, so that this was to assure that what they measured was not given by simply the accumulation of the protein fluorophore within the cells, because they really wanted to see, say, a protein that was produced and degraded in time. Okay. Then why they chose this TSR gene? So TSR gene is a membrane protein. It's a chemotaxis receptor protein with two transmembrane domains that are then fused to the N-terminus of the Venus. Okay, and why they put here a membrane protein? Because essentially, since they wanted to perform, they perform measurements at the microscope, so they really visualize cells in time. Imagine if this protein was something that simply diffused on the cytoplasm. On the cytoplasm, the diffusion coefficient is much higher than on the membrane. And so it would have been very difficult to follow the protein just because it diffused very fast. That's why they decided to focus on a protein that has a membrane localization, a membrane protein. Okay, because on the membrane diffusion is much slower. Okay, so they have this construct, and what they did was then to integrate this construct into the chromosome of E. coli. So they create a new E. coli strain called SX4, in which there is a single copy of this TSR venus gene instead of the laxate gene. Okay. And then ideally what happens is that, okay, this is the gene within the chromosome, okay? If the LAC repressor is not there, and this is something that happened, not that it happened rarely, let's say, so that it is really possible to see the transcription of the gene TSR. Okay, so when the repressor is not bound to the LAC promoter, then the gene is transcribed. We can have RNA polymerase that binds to the DNA, that the transcription begins, that we can have many ribosomes that attach to the messenger RNA and begin to translate the protein, the polypeptide, which then localizes on the membrane. And so here on the membrane, the protein is fluorescent, okay? And so by doing a time-lapse experiment, by doing a movie, they can follow the fluorescent spot. Okay. One more thing, we don't delete the endogenous lactate gene, simply say sorry, not the lactate, they don't delete the endogenous TSR gene, which is normally expressed by E. coli. As I told you, there is a gene that is a chemotaxis receptor, so it's needed by the cell. They don't delete the endogenous gene just because it is expressed in such high concentration that one more copy of TSR plus Venus doesn't change too much the function of the cells, the functionality of the cells. Okay. So now here in this figure you see, say, an example of what they got from the experiments. Okay, here you have a so-called DIC images, so a differential interference contrast image, in which we can really appreciate the presence of two E. coli cells, one here on the left and the other here on the right. Okay, and then at the same time they have also a fluorescence image corresponding to this, in which, as you can see, there are different spots, fluorescent spots, and two of them are of greater fluorescence. Okay, so we see two diffraction limited fluorescent spots only on the left cells. Okay, and what they did was somehow to attribute each of these two peaks that we can visualize here. So here we have basically the trajectory, no, not the say the intensity of the fluorescence along this yellow line. So as a function of the position in micrometers. Okay, this dashed line here marks the cellular autofluorescence. Okay. And so in what you can see is that actually corresponding to these two spots here, we have really two peaks in the fluorescence signal, in the fluorescence intensity. And what they say is that these two peaks are attributed to two different single molecules of this TSR Venus. They say this because the signal abruptly disappears because of photobleaching, upon photobleaching, which is a characteristic of single molecules. So they were really able to say, okay, we see these two spots, and these two spots are given by two single molecules of TSR Venus. Okay, so what they did was then to follow these time traces during cell lineages. Here you see another example of what they did. So they did real-time monitoring of the expression of the TSR Venus. Here you see a cell, and then after a while the cell divides, and tack, suddenly there are two yellow spots, so two single molecules, and then time passes, time goes on, and cells divides and divides, and somehow we have these yellow spots. So the TSR Venus plus Venus protein that localizes on the membrane becomes fluorescence, and they can measure it. Okay, so they could really quantify the amount of protein, the duration, so for how long these spots are present, and how many molecules belongs to each of these spots. Okay, so what they did was basically to follow time traces along more or less along cell lineages for more or less 60 traces, or more than 60 traces. So they had more than 60 different traces of these yellow spots in time, so that they can do some statistics on it, and they found different qualitative features. Different qualitative features. So features found that actually protein molecules are produced or generated in bars. And here you can actually read this. So these are the number of proteins produced, they measure, and here we have the time. So you see that the number is quite small, and its presence only is not continuous, so it's produced in bars. Then qualitatively, they could also measure the number of proteins in each burst, and saw that this number varies a lot from one burst to the other. And then the other things that we can appreciate from this plot is that the bursts exhibit particular temporal spreads. Okay, so they are not continuous. There is some temporal spreads between the bursts, which is peculiar. And then starting from this qualitative observation that they had, they could somehow answer to 4 different questions. So the first question is, do gene expression bars occur randomly? So basically, they show that the distribution of the number of gene expression bars per cell cycle for all the cells, which is exactly this, is an histogram, which is well fitted by a Poissonian distribution, okay, which is exactly this. So here they show the distribution of the number of gene expression bars per cell cycle for all the population of cells. So since this is fitted by a Poissonian distribution, then these results suggest that gene expression bars occur randomly, and most of all, they are uncorrelated in time. Okay. And then the mean of this distribution is 1.2 gene expression bars per cell cycle, so very few. Okay. Then the second question they could answer is how many messenger RNAs are responsible for each gene expression bars under the repressed condition. Basically, they could determine the average number of messenger RNA molecules per burst, M. Then they basically found that the messenger RNA molecules per burst M is equal to 1.14 plus minus something. So basically, in the repressed condition, each gene expression burst results from one messenger RNA molecule, so a single messenger RNA molecule. This implies that the lac repressor rebinds very quickly to the exposed operator region of the DNA, that allowing transcription initiation of one messenger RNA molecule. Okay. Then what they did was to fit this distribution. So here they have the distribution of the number of fluorescent protein molecules detected in each gene expression burst. Okay, this distribution is well fitted by a geometric distribution, which is this solid line here, and whose expression is this one, or also rewritten here. So this gives us the probability of generating a protein molecule from one messenger RNA. Okay, and this probability is equal to rho, which is the probability of ribosome binding, and messenger RNA molecule of the protein molecules, okay, times 1 minus rho, which is the probability of RNase E binding to the overlapping sites of the messenger RNA. Okay, so and this histogram here and the fact that it's well fitted by this geometric distribution allow the author to answer to a third question, which is what is the distribution of the number of protein molecules in each burst. And so actually they found that since the distribution fits well with an exponential decay, which basically means geometric distribution for integer n, then they could really measure the distribution of the number of protein molecules in each burst. So this distribution arises from stochastic cellular lifetime, from stochastic cellular lifetime, the lifetime, say, of the messenger RNA, which is in this case is about 1.5 minutes, due to degradation by ribonuclease, which is this RNase E that I recall you here, which competes with ribosomes for messenger RNA binding. So basically the assumption here is that there is competition for messenger RNA binding between the RNase and the ribosomes. This is well summarized by this function that fits the data. So this is a model which is consistent with the measurements that they did from their experiments. And so they could also somehow move forward and measure this. They actually fit the data, so they could measure this parameter, this probability rho. And from the fit, they found that this rho is equal to 0.8, and that the mean amount of proteins per burst is about 4.2. So this number multiplied by the number of bursts per cell cycle give us the abundance of protein at the steady state, which is more or less 5 protein molecules per cell in a cell population. Okay, this is so these results is something that they could somehow infer from the fit. They could evaluate from the fit. Then they counted the molecules at the microscope, and they found that the counted molecules are 4 plus minus something. So the two measurements are compatible one with the other. So everything is consistent somehow. Okay. And then the last thing is that the last question they answer is, what is the origin of the temporal spread of the individual bars? Okay. And this temporal spread of the individual bars can be characterized by the autocorrelation function of the fluctuations in the protein expression, which is exactly this function here. Okay, so they measure the autocorrelation time, and here the dots are the measured values, and then they could fit with a function that takes into account the average rate of expression bars, excuse me, again the probability rho of ribosome binding, and the rate constant for the TSR Venus assembly process. Okay, so, and that's it. These are the results that they found. So basically, that protein. So just to summarize, that protein molecules are generated in bars. The number of proteins varies the number of proteins in each bars varies, and they could actually measure the mean amount of proteins per each bars. And then the bars exhibit particular temporal spreads, and they could also measure it. Okay. And then now I present you the second seminal paper, so stochastic protein expression in individual cells at the single molecule level.
Hey,
okay.
Carla,
here, yes?
Would you like to take a short break?
As you want. I mean, I think I can continue, otherwise 15 or 15, 20 minutes more, and then I'm done. As you.
Okay, let's continue.
Is it okay for you all? Yeah. Okay.
Yeah. Okay.
Okay. Okay. So in the second paper, again, they could measure the protein barstiness, but with a completely different approach. Okay. So basically, they develop a microfluidic device. So they have this microfluidic based assay that allows, again, real-time observation of a fluorescent molecule, which in this case is the beta-galactosidase. Again, the model system were E. coli, so they could measure beta-galactosidase in living E. coli cells, again with a single molecule sensitivity. And then they show that their method is valid also in case of budding yeast and of some embryonic stem cells, mouse embryonic stem cells. Again, here, what they found is that protein production happens in bars with the number of proteins per bars that follows an exponential distribution. So basically, the results are exactly in agreement with the results published by the other group in the first paper that I presented you. And then they also show that bar size and bars frequency can be determined directly from real-time monitoring of the protein production. This is one thing, but can also be extracted from a measurements of the steady-state copy number distribution in a population of cell. Okay, so these are the two main results. So here the experimental setup is different. Okay, they have this fluidic assay, in which you see that basically there is a flow channel in which the cells can flow. And then thanks to two control channels or two valves, somehow cells can be stuck into one little chamber, which is a closed chamber, and they stay there. And they can, then they have a motorized stage on the microscope, so that this is the chip that they had, which is produced in PDMS, which is a specific material for producing this chip for microfluidic devices. And thanks to this motorized stage on the microscope, they could analyze more or less 100 of different chambers with cells in a single experiment. Okay, so why the need of this chamber? So and what was their experimental strategy? So basically, beta-galactosidase is an enzyme. That is only active as a tetramer. And a single molecule of this enzyme, beta-galactosidase, can produce a large number of fluorescent molecules, product molecules, okay, by hydrolyzing a synthetic fluorogenic substrate, okay, that you see here, okay, leading to the amplification of the fluorescent signal. The only problem is that these fluorescent products is not retained by the cell, within the cell, because due to efflux pumps on the cell membrane, the product is somehow expelled out of the cell, okay. And that's why they needed this closed chamber, because basically the fluorescent molecule that they want to measure it is not contained within the cells, but then after a certain time becomes to diffuse in the environment in which the cell is present, because the cell expelled this product because it's just too much, so it expelled it. And so they needed a closed chamber in order to contain the fluorescent signal and measure it, okay. And so they elaborate a sort of say test to check if really the fluorescence that they measured within the chamber was consistent with the measurements of this beta-galactosidase produced by the cell, because this is what they wanted to monitor, okay. So to circumvent this efflux problem, they trap the cells in these microfluidic chambers, such that the fluorescent product, which is expelled by the cells, simply accumulate in the small volume of the chamber. In this way, recovering the fluorescent signal, which is due to the enzymatic amplification. Okay. And these are, some tests that they did in order to see that what they were observing, the signal that it was accumulating in the chamber, was really the signal due to the beta-galactosidase. So what they measure is that the fluorescein concentration, so the fluorescent signal that they wanted to measure, increases with time in the chamber, saying that it is really accumulating. Then these lines have different discrete slopes that basically gives a measure of different auto-hydrolysis of the beta-gal molecules. And then here there is the histogram of the hydrolysis rate measured in 48 different little chambers. So basically these peaks are attributed to 0, 1, or 2 enzyme molecules per chamber. And this distribution, the total distribution, so the black line, you should look at the black line. The total distribution is well fitted by a Poissonian distribution, which is exactly this black line. Okay, and so here again, the lac promoter is important because the beta-galactosidase, beta-gal, is expressed from the LAC-Z gene, which is a gene under the control of the LAC promoter. So again, this is a gene that is expressed only when the LAC repressor is not bound to the DNA. Okay, and so in their experiments, what they did in this microfluidic experiment is basically they inject cells, okay, but they also inject the enzyme plus the substrate at the beginning of the experiments. Okay, so when they wanted to test if everything was working, they injected also the enzymes and the substrate altogether. Okay. Anyway, they have these cells. The cells are trapped in the chamber. They check that they could really say something about the enzyme amount by measuring the fluorescent signal that is accumulating in the chamber. And they perform at this point quantitative real-time measurements of individual protein expression events in all these live cells present in the chamber. And here, what we can see is that the fluorescein, so the fluorescent signal as a function that they were measuring as a function of time for a particular chamber. So this is an example of one particular chamber that contains cells that are dividing. And so what you see, what we can appreciate is that this traces shows some really sudden change in the slopes, okay, in their slope that corresponds to dividing events, okay. So corresponds to to the cells that is dividing, okay. And the red curve here is the trace of an empty chamber. So it's the reference value for an empty chamber. They measure a sort of background, and for a chamber with cells, they have something which is above the background, and that changes when cells divides. And then basically they took the time derivatives of these traces, and then they build the trace that you can see in figure B, which actually represents the number of the beta-gal molecules. And you see that corresponding to each, changes in the hydrolysis rate that we see here, there is a bump in a discrete jump in the beta-gal molecules. Okay. So the beta-gal molecules are produced in jumps somehow, okay, or in bursts again. And here in C, we see the histogram of the copy number of the beta-gal molecules per burst. And again, this distribution is well fitted with an exponential function, with an average of 5 proteins per burst. And again, is a consequence of the exponential cellular lifetime of the messenger RNA. Okay. Okay. And then, okay, so these were measurements taken from real-time, quantitative real-time experiments. So they follow in real time the traces of each single cells within the chamber in the microscope. But then they could recover some results also looking at the steady state distribution of a population of cells. Okay, the steady state distributions were distribution taken from other kind of measurements, so measurements mostly done, I think, at the cytofluorimeter somehow. And then again, they could somehow recover the steady state protein copy number distribution in a population of cells. Here you see the histogram of the hydrolysis rates for, again, the chambers that we saw before. Here we see in figure B, there is the copy number distribution of the beta-gal molecules in chambers containing only one single E. coli cells in the repressed condition. And here again, they could measure the burst per cell cycle and so the frequency of the burst, so the amount of burst in a given period of time, which in this case is the cell cycle, and the amplitude of the burst, so the mean amount of molecules per burst. Okay, and that's it. And then basically they say that they could observe, they said that they could observe two different regimes of stochasticity in the protein expression. So again, protein molecules are produced in burst, but then there can be already some existing molecules in the cells that they are measuring. And so these protein molecules produced in burst are added to the existing molecules that are these blue traces that I guess you can see here, sorry, and diluted upon cell division, leading to a given steady-state distribution of protein copy number within the cell population. Okay, and then depending on some parameter of this distribution, basically they could have say with this, when this parameter is smaller than 1, this probability peaks at protein copy number X equal to 0. And so there is a large fraction of cells that do not contain a single copy of the protein, and this is independent of the bar size. And the repressed lac operon is well described by this regime, which is exactly what they saw here. Okay, this one. Oh, this one here in E. coli. Here they repeated the experiments in Saccharomyces cerevisiae. Okay, and the other regime is when this parameter alpha A is bigger than 1, and so most cells contain the protein at some level already. But then both regimes can be described by a gamma distribution. Okay, so basically what they found is in agreement with the previous paper, and so that's why burstiness in protein production was demonstrated experimentally, was measured experimentally. Then, just to quickly conclude, what about transcriptional burst? Okay, so far we focused on protein production, on the fact that proteins are produced in, say, in burst with a certain mean amount of molecules that say allows the production of a given amount of molecules, and bursts that are produced at a given frequency during the cell cycle. But then in order to describe it, we had a model in which we have a messenger RNA molecules that is basically produced with a constant rate, assuming that the promoter is always active or is active most of the time. And then the burstiness in protein production derives from the fact that the messenger RNA molecules are amplified during protein translation. Okay, to get hints about transcriptional burst, we should now include some information about the promoter activity. Okay, so we cannot simply say promoter is always active and that's it. And there are different models that have been postulated in order to explain transcriptional bursting. So one of these is this two-state bursting model that you see schematized here. Okay, here you have a piece of DNA, a sequence for the answer, some transcription factors that are there around. The state is inactive and polymerase is detached from the DNA. And then there are two effective rates from which we go from this situation to the situation in which, say, transcription factors are attached to the enhancer. Polymerase can attach to the DNA and the transcription starts. Okay. And the switch between the inactive and the active states happen with two effective rates here called K-on and K-off. So basically the two-state bursting model suggests that a gene can flip between active and inactive states with these two rates. And only when in the active states RNA transcripts are produced at a given rate, which is here called U and degrade, or mu and degraded with a given rate, which is here called delta. Okay, and here we see a schema of the bursting parameters. So we have the off state and the on state, and here we have the trace in time, the bursting time, let's say. And we see the amplitude of the burst, the duration, and the frequency over a given time period, which is usually the cell cycle. Okay. Then there's another model, which is a slight modification of this one. So it's a modified two-state collision model. Okay, in which basically it is suggested that the intrinsic interaction between RNA polymerase and DNA are per se sufficient to create a transcriptional burst. So polymerase arrest and then followed by collision and rescue allow for individual bursts to occur. And then there's also an alternative third model, which is the so-called three-state bursting model, in which basically the data box plays a role in the game. And so this model suggests that a slow burst cycle and fast burst cycles are controlled by the binding of the data binding protein, TBP, to the promoter. And then is involved also in the subsequent recruitment of the coactivator mediator here, thus allowing a gene to be in either a non-permissive, permissive, or active state of transcription. So this is a more complex model that again can explain transcriptional bursting. Okay, so for today I'm done. Next time I will talk about transcriptional bursts and again how it was shown that actually transcriptional bars are real. So how they were measured experimentally. Okay. So I stop sharing my screen. And for today, I'm done.
Carla, thank you for your sharing.
Thank you to you. Yeah.
Sorry, I was late because I was having a meeting before that.
Don't worry.
Okay.
No, thank you for joining anyway. Yeah. Okay, so I don't know if you have any question or I can answer, or up to you. Otherwise,
that's great. Thank you.
Thank you. Okay, I'm just reading. Thank you to you.
Yeah,
I'm
the
just reading
sound
a
was
question.
a little annoying. Yeah.
Yeah, I can also hear a sound, so probably there's someone with the.
But if largely okay, I think. Yeah.
Okay, so there's a question on the chat. I apologize because when I share the screen, I cannot see the chat. I cannot I cannot see what you write in the chat, and so I'm realizing now that you were also asking here on the chat to have a short break, and I apologize, but
No,
sharing the
because
screen,
you're.
I have no access to the chat, so
That's
yeah.
fine. That's fine. No, I mean the previous message was mainly for host for Ziqiang. But anyway, I see someone else asking. Does that person want to ask him or herself?
Yeah. So now I'm reading the last question. So as someone, I read the question, as someone who doesn't understand distributions very well, can you elaborate a little on the moment generative function? How are they significant? So they are significant because, so basically they are a useful tool that we can use in order to extract moments of a distribution, from a probability distribution. In our case here is a tool basically that we can apply. Okay, it is called moment generating function because generates moments of a given distribution. And so how I show you very quickly in these slides, we can basically evaluate the moments of a distribution by subsequent derivation of the moment generating function. And that's why they are significant. Yeah, because then you go from an equation in terms of probability, which is as I also told you last time, often quite difficult to solve, to another equation which is much simpler to solve, which is rewritten in terms of the moment generating function. I would say that here is a tool that we use and that is very useful in order to get information about mean values of the molecules we are interested in and about their fluctuations, or about their, for example, we can also extract information about correlation coefficient between molecules, simply extracting more moments from the distribution, or combining the moments together. I hope to have answered. Anyway, when I will surely I'm not sure I will do this in the next seminar, but probably in 2 seminars or so, when I will talk about when I will talk about the say micro RNA mediated regulation, I will show you some specific examples in which we can apply the moment generating function to some very specific examples and see how we can use them. Okay, so another question. Is the bursting duration, does bursting duration differ from species to species from protein? This is a good question. I think so, because actually the burst duration, so the burst frequency depends on the also on the half-life of the protein, right? So depending on how the protein is stable, the burst duration should change. Okay. And then this also depends on the activation rate of the promoter somehow, and we will see how this burst frequency depends on also on the on and off state of the promoter, which which then depends on the state of the on the chromatin state of the DNA, or on the accessibility also of the answers. And so yes, I'm sure that depends that differs from species to species of proteins. Yeah.
So, Carla,
Yeah.
for this question, may I answer this way? That for different proteins expressions, I mean, in terms of the transcriptional regulations on different, even on the same protein or same mRNA, it may have different promoters in controlling
Sure.
the expressions. So maybe is it isn't the reason that bursting functions are different for that? The bursting durations are different from
You say
species to
because
species. Yeah.
yes, surely this is also no. I'm
Okay.
sure that this is also
Yeah.
another reason.
Yeah. Thank you.
Yeah, you're welcome. You're welcome. So, in a more complicated model, how can we define burst frequency and size? So, do you mean? I guess that in a more complicated model, you mean if you take into account, I don't know, protein maturation, protein
Yeah,
or
yes.
transcript elongation, and so all the details that we simply skip with this simple model. I think that one possibility could be to write down the equations and see how we can somehow map the parameters of one model to the others, and from that we can have some hints on how to redefine the burst frequency and size in terms of the other details that we decide to take into account.
Yeah, for this kind of question, I mean, in my experience has been, I think we can ask centers to tell you also. It's pretty much, sometimes we pretty much want to go backwards. I mean, how much noise do we want? And then we just go back to see, okay, with this kind of noise sound, can we define certain burst frequency and the parameters such that it pretty much makes sense, basically. Yeah, that's...
Yeah.
I guess in practice, the more important question is, because these are intrinsic noise, right? Intrinsic uncertainties. And if I want to, because a lot of times we want to map to the experiments, and we really don't know if the experimentalists are not providing us any other hints. We really don't know what kind of extrinsic noise to put on either. So I don't know. I mean, when you talk about experiment next week, maybe you could also let us know, I mean, how you consider these factors.
Yeah.
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 Sounds good. Yeah.
Thank you.
Thank you very much.
Yeah.
Okay.
So Carla,
yeah.
what do you think for our next seminar?
Yeah, so I have to apologize with you because I didn't reply to your email about the next. No, because I was checking, since here now it's a period of exams, and so I'm quite busy, and I needed to check when I had the exams and so on. That's why I didn't reply you yet. So for me, actually, it could be good again in 3 weeks. I'm sorry for this. So in 3 weeks would be I think the 30th of July. And
I'm
then
not
eventually
available on that day. How about the first week of August?
the first week of August for me is fine.
Is it? Yeah.
Yeah, it's fine.
So
then first week of August, because then the point is that after from the second week of August, I'll be on holiday, because also here there will be the summer break after that. And so in August, after the first week, I'm not here for the month of August. So I could up to you. I could do 2 seminars the first week of August if you want, or one on the first week of August and another one the last week of July, not on Friday. If you're available another day, for me is fine for the moment. But you can also tell me. I mean, you can discuss and tell me later.
I would discuss with Ziqiang and then
Yeah.
and reply to you.
Yeah.
Okay.
It's okay. Okay. So say my constraints are that I'm really busy with exams here at university until the 23rd of July, and so I'm available. Yeah, I'm available the week of the 26th, let's say from the 27, 28, 29 and 30. I'm available, and also the week after.
Okay. Okay.
Okay.
Mm-hmm.
And then you discuss and you let me know.
Mm-hmm. Okay.
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 Okay. So
Thank you very much for today.
yeah.
Thank you,
Thank
Kara.
you. Thank you.
Thank you very much.
Thank you. So let's end today's seminar. Thanks for everyone's coming.
Yeah, thanks to all of you for being patient with me. Have a nice day
Okay.
and a nice
Have a nice
weekend.
day.
Thank you.
Yeah. Okay. Thank you. Bye bye.
Bye. Thank you. Bye.
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 亲切哦，蔡志强，那样可以吗？那个网址，好，也第一个是我那个 section 这样，你有看到我名字吗？应该是第二个是我的部分。应该第一个说错，第二个是我的部分这样。对，里面没有第一个，应该是写，有吗？哈，好。